Welcome, everybody. This is a webinar that we're holding as part of the Children's Hospital of Philadelphia, a webinar entitled Collaboration for a Cure. And this is a webinar that's focusing on brain tumors, brain tumor biology, and some of the work that we're doing specifically to address how we can advance treatments and improve outcomes for children with brain tumors. I'd like to introduce some of the members who are going to be participating today. Dr. Jay Storm, to my right, is the Chief of Neurosurgery at Children's Hospital of Philadelphia. Dr. Angela Waanders is one of the attending physicians and laboratory investigators in neuro-oncology. And Dr. Adam Resnick is an assistant professor in neurosurgery and one of our principal investigators in brain tumor research. The areas that we're going to focus on today are going to be a summary of some of the aspects of pediatric brain tumor research, but a particular focus on the Children's Brain Tumor Tissue Consortium, a collaborative effort that we've developed in order to provide a major impetus to move the field ahead. So we're going to be discussing some of the reasons why we put the Brain Tumor Consortium together. We're going to be talking about some of the advances that have gone on in neurosurgery. We're going to be talking about some of the projects that are being advanced through the CBTTC. So let's move forward. I will begin by simply giving a background to some of the issues that we think are important in childhood brain tumors. Many of you understand that childhood brain tumors are the leading cause of death from childhood cancer. For those patients who do not die from their tumor, many of the survivors will actually suffer pretty significant side effects from either radiation therapy or chemotherapy. And the approaches that we have taken up to the present time have pretty much been maxed out. So we really do need to develop new therapies that will either work with existing therapies or hopefully supplant them. Some of the problems that we face in childhood brain tumors that are different from adults is that no single tumor represents a majority. As you can see from this pie chart, there are many, many different subtypes of brain tumors, and within these categories, within these slices, there are subtypes and subtypes. So we're dealing with many different tumor types in children. And the outcomes for these can be extremely different. So when we look at some of the areas that we have traditionally made very good progress on and had very good outcomes with, which include craniopharyngiomas, astrocytomas, and germinomas on the far right of the slide that you're looking at, in which in many circumstances our survival rates approach 100%. We also have examples of tumor types in which we've made very little progress. And probably the best example of that has been the diffuse intrinsic pontine glioma. And to a certain extent, some of the changes with respect to glioblastoma and the sort have a great deal of opportunity for progress to be made. We know that the overall incidence over the course of the last 30 years of brain tumors appears to be increasing. But when you look at the rate of mortality, we would hope that it would be continuing to drop as we made progress. But in point of fact, since 1990, the mortality rates for brain tumors across the board have been really quite stable, which means that we have an additional impetus to make some progress here. If you understand at this point that we need to evaluate these tumors in terms of finding out new targets, the possibilities and the combinations of these become incredibly complicated. And the point of this slide is to emphasize complexity. That this slide indicates the different ways that one signal transduction pathway, specifically through epidermal growth factor receptor, activates many different pathways on the way to transferring information to the nucleus. It could be the MEK pathway, the RAS pathway, the STAT pathways, as well as a variety of other potential targets which interact across the board, so that there are many new areas that we need to understand. If we're going to understand this, we have two fundamental choices. We can approach this in the way that we've done with cancer drug development over the course of the last 60 years, which has been to guess. If we're not going to guess, we have to have material on which to base reasoned, tactical approaches to these tumors. That involves getting tissue, that involves evaluating the tissue, that involves understanding the biology, understanding the genetics, and understanding the way that tumors require changes in order to grow and continue to expand. So I'm going to move from here to a discussion with Dr. Storm about the neurosurgical strategies which are required in order for us to even begin to approach the question, can we get to tissue? Jay?
Thanks, Peter. So I'm Jay Storm. I'm the Chief of Neurosurgery at the Children's Hospital of Philadelphia, and over the last several years, we've really put together a good team of analyzing these tumors. And it really starts with neurosurgery, and we're the ones who get the tissue. And it's important for us to begin that process of the pipeline. You have to have complete buy-in and support of everyone on the team, but it does start with us. So the neurosurgeons are where it all begins. And when we start talking about neurosurgery, we start with Harvey Cushing. Neurosurgeons love to talk about history, and Harvey Cushing is recognized as the father of modern neurosurgery, and all of us can kind of trace our lineage in neurosurgery back to Cushing, both with adult, and even here's Cushing later in his career after operating on a child with a brain tumor. And when you start looking at the way things have changed for us over the years, and this is what they started using back in 1919. This was really, up until the late 70s, the only way neurosurgeons had for evaluating the brain. They would stick a needle in the lumbar spine, drain a lot of spinal fluid out, and then inject air into the brain to try and see where pathology was. This would be normal on this side with normal ventricles, and then abnormal. But it's really amazing how several decades went by with very little improvement there, whereas now, once they would go and then go to the operating rooms, they had limited imaging, and then here's a picture of Cushing doing an operation with no magnification, no headlight, very limited access or instruments. Whereas now we have MRI scans, which give us a fantastic view of the brain. This is a normal, sagittal, axial, and coronal. So it has really, the non-invasive imaging has really changed what we're able to look at for preoperative planning and what we can do. And here's a patient, again, that would be very difficult to probably assess even a few decades ago. Now we get a very good look at this pineal region tumor. They have an obstructive hydrocephalus, and the treatment has completely changed with equipment, tools, and for us moving to more minimally invasive techniques. What Cushing and his colleagues would do would be an open procedure. You have to get in and do a craniotomy through a big opening, needles, big scissors, and again, this is all without the advantage of magnification and better access, whereas now we're able to do it through a small incision, less than a 2 centimeter opening. We can put a camera down, see into the brain itself, look around, and treat this disease safely and minimally invasively. And then after you treat the hydrocephalus, you can go ahead and biopsy the tumor all through this small hole. This is what we see when we place the camera in there. And the endoscopy over the last, for intraventricular surgery and then skull-based surgery, has really changed the way we practice operating on these patients. And this is still one of my favorite views of neurosurgery, is right when you pop into the ventricle, you get this pretty incredible view of being inside almost the center of the brain, looking at the different anatomy. And then here's where we would again be able to make this tiny hole that through a 2 centimeter opening to treat the patient and then get tissue as well. Also looking at endoscopy, again, back to what Cushing and even recently, you would have to put a speculum, and as you get deeper to a tumor in the cellar region, everything kind of cones down, whereas now with endoscopy, we can actually work through the nose, get a great view of both the anterior skull-based cellar down into the clival region to remove these tumors. This is a patient with a large craniopharyngioma that's completely solid, which would be an extensive craniotomy to resect this, where we were able to do it just through the nose to resect it and get a lot of tissue that you want to preserve. As again, we are where it all starts as the neurosurgeon, so it's important that we save as much tissue as we can and then send it off to pathology to be processed in the right way. And so this is the post-op scan showing that the tumor's been completely removed all through the nostril. There's no incision, very limited pain. This is just some fat in there to reconstruct where the tumor was removed. So as you can see, we have a lot of advances and we have plenty of tools to help us. We have the navigation that we can bring into the room. We have microscopes, endoscopes. We're able to now monitor to help us stay out of the motor strip, functional MR, where's language and motor, intracranial EEG to help narrow down exposure and resection, and much improved anesthesia, neuroanesthesia, to allow us to do awake craniotomies and better monitoring. So you can see that through all this we've come quite a long way in what we can do with neurosurgery. Maybe, because if we go back and are critically examine what we've done, we really have not done any better neurosurgically in long-term survival than we did when Harvey Cushing was operating. If you look at, as Peter had highlighted earlier, we have some tumors that, despite all our best efforts, and we can get the entire tumor out, but a lot of them will come back. And what improvements we have made that where Dr. Phillips showed moving to the right there with better survival as an outcome is a trade-off. We're using very toxic chemotherapy and especially radiation. So while we can move our survival and even cure in some points, it comes with a cost. And in the young brain, you have significant IQ problems. In the very young, we're not even able to radiate. And the key to all this is we need better understanding of the molecular biology of these tumors. And that was something that a lot of us have recognized for a long time, but it can't be done by an individual surgeon, can't be done by an individual neuro-oncologist or an individual surgeon. It can't be done at one institution, especially for pediatrics where the numbers are so small. And when I got to CHOP 12 years ago, it was with the understanding of being able to recruit people who are like-minded to work with us, who are at CHOP. And in the case of Adam Resnick, who you'll hear from later, was someone who I brought with me a couple years after I got to CHOP. We'd worked together at Johns Hopkins and had a friendship and a collaborative work, and that's what's continued here. And when you look at our team here, these are just some of the people that make this work. And so we need the neurosurgeons to start. We need pathology. There's Rita Santi, who has been a fabulous partner in helping process. The patient then moves on to our oncologists like Angela Waanders and Peter Phillips. And as they're working on clinical and basic science work, we also have partnered with Adam Resnick, and that's kind of like our core group at CHOP now, amongst many others. I mean, this all branches out very quickly. And not only have we done that locally, but as you'll hear in our next two speakers, we've now kind of got our house in order locally, and they're now branching out throughout the country and actually now throughout the world to try and recruit other members to work with us and to get the volume up, because that's what it takes to do the right kind of science. And technology now is such that in order to avail ourselves of this rapidly changing landscape, we need partners and we need the volume, and that's what we've been doing. So at this point, I will turn it over to my oncology colleague, Angela.
Are we going to break for a few questions?
So we're going to start with our first questions. The first question that's been posed to us is, in what direction do you see the treatment of brain tumors going in the next year or two? Dr. Waanders, do you want to make some comments about that?
Well, I would love to be able to say that we are going to make significant innovations within the next year or two and move the treatment or the overall survival curve. Realistically, I don't think that the actual treatment is going to change significantly within the next year or two, but I do think that the biology and the research discoveries that will get us to better treatments is going to change significantly in the next year or two. So I would change that question a little bit, say we're on the cusp of making some very important discoveries that will translate into better treatments within the next 5 to 10 years. For my patients who are out there, my patient families, I know that this sounds like a long road ahead, and I'll highlight when I talk a little bit of why we must first understand the biology and not just move into treatments right away. Otherwise, we could do more harm than good.
Thank you. Another question is, do you feel that things are starting to change in terms of the focus of funding and awareness in the area of pediatric brain tumor research? Adam, what do you think?
So I think that we face multiple challenges for doing the kind of science that we need to do in general, whether it's for pediatric or adult diseases. Especially over the last several years, funding rates from the NIH, the main body that provides funding for academic and basic research, have dropped or stayed stagnant. And universally, across time, the pediatric entities that were applying for funding have always been underrepresented for multiple different reasons. And so the rates of funding or the availability of funding from the traditional sources like the NIH, those I don't think are poised to change dramatically in support of our efforts. But I think through efforts like this and advocacy and dissemination of information, I think you can see a coalition of patients, their families, and foundations who have taken ownership of the space of supporting this kind of research in ways that are authentically empowering what we do on a daily basis at CHOP and across the country and elsewhere. And so I think that's the movement that's changing, is the interactions with the families and foundations in ways that have empowered us to do what we're doing here and what we hope to do in the future.
Great, thanks. Are there new types of promising surgeries coming down the line for more difficult to get to tumors? I'll do that one.
So the
I think that a lot of improvements with surgery are with instruments, both as we start doing more and more of the endoscopic skull-based surgery. A lot of that, especially in pediatrics, is just becoming more popular over the last several years. And we're using a lot of the instruments that were designed for the otolaryngologists and sinus surgery, and a lot of them are not amenable to some of the surgery that we're doing. So we're always, as we're doing these new surgeries, we're always working with some of the companies to work with better instruments. We have endoscopes that can look 30 degrees, 45 degrees, but we have 0-degree instruments. So occasionally you can see a tumor around a corner, you just can't reach the tumor around the corner. And I think that as we've seen, as I was talking about with the presentation, we're moving towards getting just as good of results with being able to do it through smaller incisions, which means shorter hospital stays, I think less discomfort for the patients. And there are even some tumors, again, that I think we can treat through, there's some new lasers that are coming out that I always find interesting, because for years people have always said, can't you just use a laser for that? And now actually we can start to say, yes, actually there are lasers that can be used for certain surgeries, both for epilepsy and maybe deep tumors that we would otherwise just watch. Maybe now we could biopsy them and just slide a little catheter in to laser them. So most of the advances, though, are going to be through equipment, and the limit for most of us taking out tumors is the location, because of the structures that are so important. And I think, again, that is what we need to treat. And the real advances surgically are going to be the ones that are not made in the operating room, so that we can treat the tumors that we can't completely remove.
Thanks.
Can I add just one comment that I don't think you're giving yourself credit for it's not the tools, having done second opinions and seeing referrals come through CHOP. I can't comment enough it's on your judgment and your skill. So you can take a not so good surgeon and give them all the tools and all of the fancy tools that they could have, and it still wouldn't equal someone who has skilled hands and good judgment and good communication. Yeah, sorry, I just had to put that out there.
Well, there's certainly no, when I always talk to the medical students, we talk about there is no substitution for good judgment, knowledge of neuroanatomy, because, again, Harvey Cushing didn't have any of these tools, and he was able to get good results. So, again, surgical experience and judgment is certainly extremely important.
Sorry to interrupt.
Great. No, fine. I'm going to stop this question session with the last question, which would be, what can the public do to support your research? And some of the comments that I can make on that are pretty straightforward. Look, this is very complicated work that we're doing right now. It is time-consuming. It is involving many, many different people, and the technologies are themselves expensive. The primary thing that the public can do to support our research is financial. As we're going to talk about today with respect to both the Children's Brain Tumor Tissue Consortium's fundamental activities, as well as some of the research projects that are coming out of it, we have a desire to set up something that is going to move the field forward. But this takes a substantial amount of our financial resources. It's costly, and what we need to do in terms of the analyses are costly. So what can be done in terms of supporting our research would be contributions, however small, make a huge difference to us, both knowing that you're out there and behind us, as well as providing the ammunition that we need in order to make this happen. Angela, you want to do some discussion here?
So I'm going to start off by talking a little bit about background and how I became involved in this effort. I think one of the things that we often don't talk about are the stories that lead us to the career decisions and where we find ourselves. I'll be honest, 20 years ago, I did not have a burning desire to be a pediatric neuro-oncologist. Even as close as 10 years ago, I had absolutely no desire to work with mice in the laboratory and to use them as little patients. But everyone has these kind of fundamental times or crossroads in their careers and their lives. And for me, it really was in 2006 when I was a pediatric oncology, a clinical fellow during my year in that inpatient service. When you are telling families that there's nothing more to offer or that you have nothing better to offer, it really is motivating to ask the question, why don't we have something better? In terms of the treatment strategies and the treatments that we had available in 2006, I kept asking, why are we using regimens like this? Why are we choosing this specific class of medications? And it really became, these are medications that have been around for decades. We're rearranging them, trying to incrementally improve survival, but we don't actually have a basic understanding of the biology of these tumors, so we can't develop something more rational. I think it's akin to, you're on the Titanic, you're rearranging the deck chairs, but you're not actually adding any more lifeboats to it. So one of my goals and one of the things that I decided I wanted out of my career was to make a difference and to be able to say, at the end of my career, this is what we have done. And I think everyone sitting at this table and everyone involved in the brain tumor program at CHOP, whether you're a researcher or a clinician, we all have this same kind of drive and motivation. So in terms of specifics, I'm going to just kind of walk through a little bit of the project, a research project that I've been working on. So going to my comment of being a little bit shocked at the current state of pediatric brain tumor treatments back in 2006, one of the things I realized is that we treat based on histology, what a tumor looks like under the microscope to a pathologist, regardless of the patient's age, regardless of where the tumor was located. Everyone kind of gets the same standard treatment. It's not tailored, it's not personalized, it's not based on genetics or molecular alterations. So for pediatric low-grade gliomas, which is I think where we've made some significant strides, we're really trying to shift this paradigm. The MRI on your far left, or of the cerebellar classic astrocytoma, that's one where Dr. Storm and his team can actually provide a cure. For the rest of the MRI images, these are patients for which we are still using chemotherapy agents that were discovered and developed decades ago. And so we're really trying to shift this paradigm, moving away from a one-treatment-fits-all approach to pediatric brain tumors to a genomic approach. So the specific project that I happen to be on, and kind of one of the things that got me excited about brain tumor research, was back in 2007 in the Beagle laboratory. We were using what was then considered the most advanced technology for genomic screening, something called a high-density SNP array platform. So we and other groups were one of the first to discover a B-Raf fusion gene in low-grade gliomas. This was exciting because this was the first time that a genetic alteration was found in this tumor type, as well as it was potentially a mutation that we could target and that we could treat. So at the time, as a naive early investigator, I assumed we had this great project, we had this genetic mutation that we discovered, that pharmaceutical companies and the government would just be equally excited, give us their drugs, give us their money, and that 5 or 10 years later we would have cures for these patients. Unfortunately, that's not what has happened, but I think the story has been informative for us. So the B-Raf mutation that we discovered is unique at the time because it's unlike the other types of B-Raf mutations that are found in adult tumors. Part of what is unique is that rather than a single point mutation, the B-Raf mutation that we discovered in the pediatric low-grade gliomas actually has taken a part of the chromosome, duplicated itself, and created what we call a fusion gene. When we studied this fusion gene in the lab, what we discovered was that the drugs that had been designed and had already been in clinical trial in adult cancers, and that were showing great promise in melting away adult tumors with B-Raf mutations, did not work and were not effective for the B-Raf fusion that we discovered in the childhood low-grade gliomas. In fact, at the time, before we had made this discovery, there was a clinical trial that had opened up that was trying to use these same drugs that closed to early accrual because they found clinically what we were finding in the lab, that exposing drugs to this mutation actually made tumors grow faster. So really what we're trying to work on at this point is not only finding the genetic alterations or the genetic mutations, but really trying to understand the biology, the unique features, and how they drive tumor growth, so that basically we can find the appropriate switches, the right switches, to turn off tumor growth and potentially provide new and different cures for patients. What's really unique about this kind of personalized medicine approach or personalized genomic approach to treating tumors is that we're not going to use the traditional cytotoxic chemotherapies and radiation therapy. They may be additional adjuncts or things that we use in combination, but we're trying to target and treat the tumor in a completely different way than we used to be able to, say, 10, 20 years ago. This is one of my first mouse experiments in which we took one of the B-Raf mutations that we were studying. We injected flanked tumors into mice and then gave them the different drugs that we were trying, that we thought might be promising. The curve that I want you to focus on this particular slide, though, is the red line. Let's see if I can use the pointer with that. This red line right here. So these are mice that we treated with a combination drug that is not a traditional chemotherapy. And in all of the mice that we treated, the tumors not only responded, but they didn't grow. So to me, this is very, very promising. What I want to caution, though, is that this is still in preclinical staging. So while these types of findings are very exciting, you do have to be careful about moving them into the clinic and giving them to patients too soon before you understand them. So a lot of our work has been funded by foundations and by direct donor support. The particular project that I was talking about was in part funded by the Pediatric Low-Grade Astrocytoma Foundation, and has directly supported or led to a different kind of clinical trial through another consortium group that we're all involved in called PNOC. So there are now two clinical trials that are open and available, looking specifically for new treatments in children with low-grade gliomas. So this is kind of an example where proof of concept, where you find a genomic alteration, you understand the biology, you work with multiple institutions and multiple partners and collaborators, and you move something forward in the right way into the clinical trial phase. So where we are now, and Adam or Dr. Resnick will elaborate a little bit more on some of the other research efforts that we have going on, but where we are now is that technology has moved. So back in 2006 and 2007, when I was studying brain tumors initially, we had high-density SNP array platforms. We were just beginning to unravel the genetic makeup of tumors. We've now moved technology so that we have now the genetic data is there, and we now have to make sense of it. So back in 2007, we discovered one B-Raf mutation. There are now a number of different B-Raf mutations that have been described, as well as different fusion and different mutations. This is just within pediatric low-grade gliomas. And we are now just beginning to unravel if a patient has, say, this B-Raf mutation, what drug to give them versus a patient who might have a mutation in the FGFR family, the NTRK family, MYB family, or RAF1 family. So we are very much moving towards a personalized medicine approach for the low-grade gliomas and other tumor types. One of the things, just so I don't forget to mention, that we really need to move forward with as well is moving this testing out of the research testing only phase and moving it into the clinical phase. Right now, if I have a patient who has a low-grade glioma, we test for the KIA B-Raf mutation and for a point mutation in V600E. We do not test clinically for all of these other mutations on a routine basis. I personally think that's the wrong thing to do, that we need to start knowing which patients have which mutations, so then we can better understand the biology, and then when we have drugs that are ready to go, we already know which patients that will benefit from them. Other types of personalized medicine approaches that we and other institutions and collaborators are working on, for example, are high-grade gliomas, which Dr. Phillips mentioned we have not made much impact on at all in terms of survival, as well as diffuse intrinsic pontine gliomas. The Courtney Rose Foundation, which is one of our local donors, has really helped to drive that effort moving forward, as have a couple of other different direct donor sources. We also have a medulloblastoma sequencing effort project, and kind of doing the same personalized medicine approach of, once we discover the genomic and the molecular alterations that drive tumor growth, we can then design better therapies for them. So really what we need moving forward is to understand the biology, to identify the best new therapies moving forward, so that we don't make incremental changes, that we actually significantly and meaningfully move research and move treatments forward. And also how we use these new treatments in terms of clinical trials. So I don't know.
All right, so I'm the closer. I think, Angela said that she came into neuro-oncology sort of later in her career. I'm always wanted to be a scientist as far as I can remember. I went into school wanting to be a scientist, specifically a neuroscientist. And in the early 2000s, my vision in space for what I wanted to do for the rest of my life changed dramatically. And what I wanted to do as part of this presentation is to highlight what is something that is very challenging to convey, is the opportunities and possibilities associated with the scientific realm as we try to apply it to the clinical care, and how far and how fast we are currently moving in ways that, as everybody else here has suggested, really accelerates the space of both doing the research and being able to apply it in this very complex arena. So around the early 2000s, when I was still a graduate student at Hopkins doing neuroscience training, the first human genome was sequenced. And this was a tremendous endeavor that included tens of laboratories and took more than 10 years to do, at a cost of more than $3 billion to actually achieve. The graph on the right essentially highlights our acceleration and capacity to begin interrogating genomic data across time over the last 10 years or so. So when I was in graduate school in the early 2000s, it took more than 10 years and $3 billion. And today at CHOP and many other places, we can sequence the DNA associated with tumors within a day or so at a very reduced cost, in $1,000 or around there. And that provides such an amazing opportunity for us to begin really interrogating what it is that we can understand about the biology of the tumors. When all of us are talking about these genetic mutations that we find in tumors, it's important to understand that the mutations are specific for the tumors. It's not, it's very rarely something you inherited from your parents, or because of anything else that essentially you did or your parents did. As far as we can tell, most of the mutations are random events for reasons that we don't quite understand. And when all of us are talking about mutations, whether it's in B-Raf or any other setting, what we're really talking about is alteration in the code of the genome of the cell. And this slide just essentially provides an overview of the biology as instructions that's important for us to think about in the space of understanding the biology and then implementing care based on this biology. And so each one of our cells has a DNA in it that's packed into chromosome, and that DNA essentially is the master instruction set for all of life. And those instructions then get converted into functional units like proteins that essentially determine what a cell does or what kind of cell it is. So whether it is a cell in your eye or in your big toe or a tumor cell. And when we're talking about precision medicine, what we're really talking about is understanding where do the instructions go wrong. And the tremendous acceleration of capacity to sequence genomic information really allows us to identify what is wrong. Now, it doesn't mean that we have tools to directly treat what is wrong or even know what that means. But now we can identify at least what's different than what's expected. So as Angela showed, and as also Peter showed early on, this knowledge space then gets incorporated into an understanding of a signaling complex that essentially is what the cancer has altered, right? So all cancers essentially have a mandate of simply dividing and surviving. And cancers choose a number of different solutions to that problem and include a host of signaling events and hosts of proteins and messages that are altered or stuck in the on or off position. But what we're learning over the last several years as we're beginning to amass very large amounts of data is that the picture is actually much, much more complex. And the complexity associated with life itself, but also with the biology of tumors, is of an enormous nature, where even though we try to reduce this complexity in slides that simply show a few components, 11 or so in the B-Raf pathway, the 25,000 sort of potential members of the signaling complex interact and integrate information in very complex ways that require deep understanding for both affecting them when we're going to target them, but also to predict how the tumor might respond once you begin targeting them. So to engage this complexity requires actually amassing large amounts of data. And as Peter and Jay and Angela highlighted, for us, when we began initiating some of these collaborative efforts, that was the first challenge or hurdle to address, is how do we begin doing this type of research to define the landscape of pediatric brain tumors. Now, we and others, both in the adult and pediatric space, all recognize that these powerful techniques provide great venue into understanding the biology. But as the question that were asked earlier highlighted, it's often been challenging to sort of implement some of these approaches in the pediatric space because of the need for funding. But the NIH and the main bodies that sort of fund cancer research did engage in many efforts to generate data associated with tumors of all kinds. One of the main large efforts that began in 2006 is called the Cancer Genome Atlas, or the TCGA effort. And since 2006, they've sequenced the genomes and characterized more than 11,000 samples across 33 different tumor types with thousands of publications that essentially led to the definition of a large number of tumor subtypes and the causes of tumors, largely in the adult space within this effort. In contrast, over the same time period, pediatric efforts sponsored by the NIH or government entities have lagged behind. So during that same time period, other efforts sponsored by the NIH have only resulted in around 1,300 subjects being sequenced for only about 5 different tumor types, which doesn't include brain tumors. And so this highlights sort of the space of ownership that both CHOP and other pediatric institutions are trying to engage, and the space of ownership that I think we are trying to get families and the rest of the community to engage is a recognition that in the pediatric space, we're very far behind in applying what are tremendously powerful techniques to understand the biology and implement new therapies. As you've heard, the only way to do that, no matter how great a surgeon or oncologist or how big a hospital you have, is to solve the tissue access problem, to get enough biospecimens to begin interrogating this very complex biology. And so currently, the CBTTC institutions that have partnered with us, that list is growing, but that includes CHOP, Seattle, Chicago, Pittsburgh, and the University of California, with new members coming on board in the United States, and the Mayo Children's Hospital in Florence already joining in our efforts. In this collaborative space where all the institutions engage in a shared environment of addressing this great need has been phenomenal for what it is that we've begun to do with work like Angela's in low-grade glioma and what we hope to do and empower others to do. We're engaging a new economy of science in which the data and the specimens that we collect is available for the larger community to address. We recognize that there are brilliant people everywhere across all domains of science, and we'd like to engage them as deeply as possible in looking at brain tumors. Now, when we talk about a biorepository that simply collects specimens, many people default into the idea that maybe you have a freezer in your laboratory in which you just put biospecimen. For us, there is a synonymous definition between a biorepository and collaboration. Biorepository for us means a space of collaborative work that's empowered by the specimens and the families and children who essentially have participated in our efforts to collect those specimens. But still, you need freezers. And our scope of endeavors far exceeds sort of a traditional laboratory freezer, such that now we have freezers in the basement of CHOP that can house more than 2 million samples and are robotically managed. Now, the specimens there don't belong to CHOP or any other institution. We at CHOP are simply stewards of specimens trying to engage research. They are specimens that belong to the community and the families and patients that have contributed them. Specimens are the first step. But once you have the specimens, suddenly the repertoire of activities that allow you to define what is actually wrong open up. And we do this through a number of different ways. One, looking at the sequencing of tumors from both a number of different perspectives, characterizing the molecular components of the tumor, but also generating model system. And Angela highlighted it's incumbent on us to test and validate everything we find as deeply as possible before implementing it in the clinic. We cannot do any harm. And we understand how complex and variable the biology can be. And so we spend enormous amounts of time across a number of years typically to define and understand the approaches we're actually going to implement in the clinic. The biospecimen in the first place. The other thing that empowers the use of biospecimens is actually collecting all the clinical data associated with the specimen. And so we actually follow all the patients in the repository across space and time, collect the clinical data from their pathology and their treatments that they've received, and begin integrating both the clinical data and the biospecimen data, creating a database that becomes open to the pediatric community and beyond to interrogate what it is that's available for research. There's way more research than Adam Resnick can do or any one of us here can do. And so we begin encouraging, and we encourage you to advocate for additional needs for research and to inform that now the bar is a little bit lower because there are resources to do this. We do have more than 1,300 biospecimens from all of these institutions, very well annotated and available for research. Now, we've heard a lot about the genomic landscape. And again, it's one of the more difficult things to convey of the enormity of the opportunity that's associated with it. And that in the very near future, what seems sort of almost impossible, when the first human genome was sequenced, will come standard fare for almost every single patient at CHOP and other hospitals, where the genomes and the information about them, the patients and the tumors, will be universally generated on a daily basis. And that's because the techniques have improved. We now have an opportunity to understand these highly complex networks. But besides collecting specimens and clinical data, the next challenge now emerges. And that's because the genome represents a lot of data. Each genome is comprised of more than 3 billion little A's, T's, C's, and G's that essentially provide the template for information. And each time we sequence the tumor and try to integrate it, it generates a large number of terabytes and eventually petabytes of data across space of time. That ends up being both informative and complex to interpret. But another challenge is actually how to work on it and share that data. It's easy to share pictures and limited information, But it turns out that actually working and understanding the sequencing can be very challenging. This, for example, is a tiny fragment of the kind of data we get when we sequence a tumor that comes through the operating room. The pieces of the genome come at essentially 100 letter-long pieces that we stack and try to order across 3 billion letters in order to make sense of the alterations. And here on the top right, you can see that there, instead of an A, we found a G. That alteration then has to be interpreted for significance and potential implications for the cancer. And it turns out that the community, whether you're pediatric or adult, still hasn't quite figured out how to do this robustly and reproducibly. And so there's great mandate for data sharing and interpretation and integration of data. But that becomes more and more challenging the more data you have. And so in addition to engaging in an economy of science that's associated with building tissue repositories of biospecimens and clinical data, now there's a mandate for sharing this data. Yes, there's not a lot of data out there, but there will be, and we need to empower it for its use. And in one way, we're actually borrowing an example from biology itself and cancer. Cancer and biology is dependent on these very complex networks to survive and do what they do. And traditionally, the model for doing science has not been one of networked. My vision as a scientist, when I wanted to grow up and be a scientist, was a scientist working in the basement of some university alone, trying to make the discovery of a lifetime and win a Nobel Prize. But now we know that that's not the way to do science when it is in this space. Now we need to integrate knowledge across diversity of expertise and share this space in a way that create networks. And the networks themselves are actually what empower the science. So instead of having a radial approach where you are at the center and potentially disseminating your discoveries, we actually want to work together to generate discoveries. And so this large amounts of data, the next phase for our efforts, the CBTTC's efforts, is to begin sharing this data and integrating it. And we're moving into the cloud. We can, again, only do this using support from foundations such as the Dragon Master Foundation. While this is the clear answer to how to share pediatric data, currently much of the data that's being shared in this way is still adult data. And so again, it becomes incumbent on us and our partnerships with you and others to advocate for resources and opportunities to do this. And we're partnering with a number of companies to help us disseminate and share this data.
So Adam, with the kind of intense work that's necessary under these circumstances and the complexity that's involved, where are we getting the support for CBTTC? Where's the primary support coming from that allows us to continue day by day?
Right. So what Peter is highlighting that is that at each and every step that we move through these efforts, from the initial support to collect specimens and onwards, we need a diversity of partners. The initial partner that helped us set up simply the repository itself and the initial collection was the CBTF Foundation that provided that. And on top of that, we have probably hundreds.
Children's Brain Tumor Foundation.
Children's Brain Tumor Foundation. And on top of that, we have a diversity of foundations that either support projects on a specific type of disease or the repository itself. But the support, again, is not just for CHOP. We're really supporting the entirety of the community. And this is what I wanted to show in this last slide, is that as we build this network, the network now begins to get overlaid. Not only from the Children's Brain Tumor Tissue Consortium network of hospital, now we're layering on top of that partnerships in the Clinical Trial Consortium, the PNOC Consortium, in order to implement this knowledge space as we begin developing clinical trials. And that space then gets integrated into more than 300 institutions that have signed on to the Global Alliance for Genomics and Health, which is a body of people who are in institutions who recognize that in contrast to many of the domains of life that we as individuals normally interact with, where we can reach and understand almost anything through the internet, sequencing is not internet ready yet. And that's a main barrier for actually sharing data, is putting data on the internet for sharing and working on it.
Good. So let's go back to some of the questions that have been asked that may relate to some of the work that we're doing now and may relate to work that comes from other institutions in the future. Some of the questions kind of converge around an issue of immunotherapy or viral-based therapy. And one of them is that there's been so much news that's been going on with respect to immunotherapy to treat cancer. Is there eventually something that's going to be useful in treating brain tumors, and especially when is this going to come into childhood use? And I think that there's some important issues to understand under these circumstances, that viral-based therapies have been instituted, including in children, for over the last 2 decades. Viruses used to introduce new genetic material, to change the susceptibility of a tumor to a particular agent, as well as to actually be primarily responsible for killing those cells. One of the things that is critically important in advancing a therapy like this for children is going to be either to demonstrate in a similar tumor type in adults that this is safe and effective before we move it into children's use, or that it has a target which is unique to children's brain tumors. At the present time, the vast majority of the immunotherapy strategies and viral-based strategies have been targeting gliomas. And there are some interesting issues that are coming up. We do know that a number of institutions are planning to and expecting to extend these to children. But again, the safe and effective criteria are 2 of the things that have to be demonstrated in adult trials before we move into children's trials.
Right. We get a lot of questions about immunotherapy here at CHOP, because CHOP has a very robust partnership with the University of Pennsylvania for immunotherapy. And like Peter said, immunotherapy is not new, but what is new is the application of genomics. And so for us, we see the same advances that Angela talked about by having specific drugs that target a mutant protein. The next phase is the application of new techniques like immunotherapy that are based specifically on the mutation or specificity of the tumor. CHOP and a number of other institutions are leading such efforts. One such effort is called the Stand Up to Cancer effort, funded by the St. Baldrick's Foundation, which again directly tries to address identifying the unique molecular makeup of tumors based on examining biospecimens and designing immunotherapy approaches that can be implemented there. The Stand Up to Cancer effort addresses a number of different pediatric tumors, but one such subset that we are involved in is the application in the brain tumor field. But like Peter suggested, for us the mandate is to demonstrate safety. But they're beginning to accumulate data that suggests that this is an unprecedented opportunity for personalized medicine. And we're actively working to make the scientific efforts proximal to their implementation in the clinic. But it'll take a number of years for us to begin to do this as we move forward. But again, it's tied to being able to understand the biology and intersecting that with the science that you can do in the laboratory.
Great. Listen, there are a number of questions that have coalesced around the topic of diffuse intrinsic pontine gliomas. And those questions address 2 general areas. One, what's the safety, as it stands right now, of biopsying diffuse intrinsic pontine gliomas in order to get tissue for analysis? And the second question is one that we'll address is, and once we do that, is there progress that's being made in DIPG that can arise from this? You want to comment on, James, on some of the issues in safety?
I think that from a safety standpoint, it actually is a pretty safe procedure. You know, some of the ones that we've done before that looked atypical, we would do open and go in and get certainly plenty of tissue, but you can also stick a needle in there relatively easy, I think. And I've certainly been an advocate for a long time that we should be biopsying more of these tumors and getting tissue so that we can start looking. Obviously, getting a lot of tissue in that area is not something that can easily be done, but as these techniques have gotten better and better, I think some of the original concerns were you're not going to get enough tissue anyway to do much about it. These techniques now are so good where you can even get down to one cell and start looking at it, especially when we've learned so much about other tumors. Is there something in DIPG that we can target? And once you know that, then it's very easy to get a small sample, and then you have plenty of information to follow it up. So going forward, I think from a safety standpoint, it's very easy, and because of the changes even in the last few years, and even it's changing daily almost with these techniques, that we are able to get enough tissue that it does make sense that we should be biopsying these, and the only way to learn about them is to biopsy them.
Yeah. So in terms of making progress, I can make simply the statement that Angela and Adam can comment on that. Up to the present time, we can say definitively that over the course of the last 60 years we have made no progress. But there is some reason to expect and to anticipate that there are changes that are being identified that may be the fodder for and the impetus for some progress. You want to comment on this?
Yeah, I'll start first by saying that, as Jay highlighted, the DIPG space highlights better than almost any other tumor type the authentic partnership required with patients and their families, because the biopsies that patients underwent to date have been largely for research purposes. But as Peter highlighted, there's an immense body of knowledge that just evolved over the last 2 years. And now we actually know a lot about what it is that's causing these tumors. And only very recently do we have specific potential opportunities to target those. But almost like we found out in any other disease space, a term like DIPG does not actually mean one disease. And it will require for us to understand how that one disease actually bifurcates into discrete molecular subtypes and how to target them.
I think a key component of that too, in terms of our understanding, is something like what we're trying to do with the Tissue Consortium is not have a freezer full of tissue that we can sequence and know what genetic mutations are there, but to actually make it meaningful, be able to move that into the clinical realm so patients can get biopsied, can find out what mutations they have, and then also that we can track and follow over time of how do different patients in these different subgroups actually do. Do some respond to radiation better than others, and what is the time course of the disease?
Yeah, and that's the next phase of clinical trials that you'll see at PNOC and elsewhere, where the sequencing is proximal to the clinical trial implementation.
Great. One of the questions that's come up is, we all know that the blood-brain barrier prevents many drugs from effectively reaching their target. Are there advances in delivery methods that may be helpful under these circumstances, and if so, can you describe them?
Yeah, I'll start by first highlighting that it is challenging. As Angela described, many drugs that we potentially test and try to implement initially in the laboratory and then further on, many of those drugs are not initially designed for brain tumors, right? The B-Raf inhibitor was designed for melanoma. And in fact, most drug companies purposefully design drugs to prevent them from going into the brain, to limit side effects in the disease of their interest, right? So if you're treating melanoma, you would rather not have the drug go into the brain. And that presents a challenging space for us to address. And it's a continuous challenge. There are a number of groups, CHOP and elsewhere, that are looking at convection-based therapies and other types of delivery, nanoparticle delivery for tumors. And we're still advocating for drug companies to consider developing drugs specifically for the brain and encourage that development. And in some instances, we get lucky. and that a drug can still penetrate the tumor environment because of a breakdown in the blood-brain barrier associated with the tumor.
One of the complexities, though, in delivery issues is that recognizing that the brain tumors themselves, many of them, have no blood-brain barrier, right. And that finding agents which are able to kill these tumor cells, even under the best of circumstances, is a challenge. Yeah. So, for example, in medulloblastoma, the blood-brain barrier for these tumors is about as open as it is in the liver. Our challenge is to find drugs first that work, that are able to get the tumor smaller, and then at the boundary of these tumors where the blood-brain barrier may be intact, work on strategies for developing improved delivery. But it's a two-phase problem. It's a problem both of finding intrinsic efficacy as well as adequate delivery. And certainly for molecules that are extremely large, like antibodies and the sort, delivery is can be a considerable problem.
And I think that highlights too of testing some of these drugs for proof of principle in the research setting first. I know a lot of people feel uncomfortable with the thought of doing animal preclinical testing, but for some disease types and for some drugs, we actually do need to do that to prove some of these questions first.
I think we'll finish with a question. Can you comment on the results of a ganglioglioma project that was recently worked on?
So, as I mentioned, we receive support universally initially through foundations and grateful families before we can even successfully apply for NIH support. A ganglioglioma project that we did receive support from the Voice Against Brain Cancer group supported the sequencing and analysis of gangliogliomas in the context of a number of different low-grade gliomas, and actually identified that there's overlap. So between ganglioglioma biology and other tumor subtypes. So this is a reverse presentation of what we have highlighted, which is within medulloblastoma you might have 4 or 5 different discrete categories of tumors, but it actually also ends up being the case that across tumors you might have some shared biology that you need to be aware of. And I think that's important to recognize, and as Angela highlighted, it means that when you design trials, you should be thinking about it from the molecular perspective, not necessarily from only the name of the tumor. And that's a tremendously powerful space for us to explore in ganglioglioma and elsewhere.
Great. That, in closing, one last question was, is the Children's Miracle Network part of the Children's Brain Tumor Tissue Consortium? And in point of fact, the Children's Miracle Network is a major supporter of Children's Hospital of Philadelphia. The Children's Hospital of Philadelphia is probably the largest supporter of the Children's Brain Tumor Tissue Consortium, providing us with a kind of an infrastructure that could not be done at any other institution at this time. So in that sense, the Children's Miracle Network is a critical component, although our principal supporter is the Children's Brain Tumor Foundation, as well as a multitude of other private foundations without whom we could not possibly function. So I want to close at this point to thank everybody in participating, that there will be an opportunity, I think, within on a internet written response basis to some of the other questions that we weren't able to get to today. As you can see, this is a topic that we are all deeply passionate about and can talk forever about. So in the interest of time, we'll close today and look forward to talking to you again. Thanks so much.
Thank you, everyone.
Thank you.
Bye.